Welcome to our STR presentation by Kayla, Blake, Rowan, and Alexis. What is STR? STR stands for Short Tandem Repeats.
Short tandem repeats, also known as microsatellites, are short tandemly repeated DNA sequences that involve a repetitive unit. They appear to be scattered throughout the human genome and make up about 3% of the entire genome. STRs are largely found in prokaryotes and eukaryotes, including humans. Short tandem repeats were discovered in eukaryotic DNA in the late 1960s. The first model was invented in 1964 by Kornberg et al., who proposed a model called DNA slippage, which is now a popular explanation of the short tandem repeats mutation process.
In a village nearby the city of Kosovo, a married couple was found killed and quartered. During the autopsy, blood from the two victims, nail debris, and pieces from the ropes were sent for DNA analysis. Extraction of DNA was made with a mini-kit. The PCR reaction was performed with an identifier amplification kit and capillary electrophoresis was done using 310 genetic analyzer. DNA from the rope, which was used to tie the male victim's legs, matched the optimal STR profile from an unknown male. The perpetrator was then later caught at a church burglary in which DNA was also left at the scene. The previous sample, which had taken 5 years earlier and stored in the Macedonian DNA base, was a match, thus solving both cases and putting him behind bars.
Here's a physical illustration of what investigators would be looking at. They are looking for repeating patterns to see if they match with someone else's DNA. If the pattern matches up, it could mean that the DNA samples are from the same person or related individual. Short tandem repeats are used in forensic analysis primarily for DNA profiling. They are particularly useful because they are highly polymorphic, meaning they vary greatly between individuals. This variability makes them invaluable for identifying individuals in forensic investigations, such as crime scenes or paternity tests. STR analysis is especially effective when only a small or degraded DNA sample is available. Short tandem repeats are essential tools in forensic science. Thanks to advancements in technology, analyzing these repeats has become faster, cheaper, and more accurate. This means forensic labs can quickly analyze DNA samples for crime scenes, helping law enforcement catch criminals and bring closure to victims' families. So the use of STR today is a big deal in forensic investigations, playing a crucial role in modern crime-solving efforts.
Short tandem repeats analysis involves 3 key processes: amplification, electrophoresis, and interpretation. In this step, the DNA sample is mixed with reagents and heated, causing the DNA strands to separate. This separation allows for the creation of millions of DNA copies, making it possible to analyze even the smallest DNA samples. Following amplification, the DNA fragments are separated based on their size and electric charge. This is achieved by placing the DNA samples in a gel and applying an electric current. The smaller fragments move faster and thus travel further than the larger fragments, allowing for effective separation and visualization of the different DNA fragments. The final step involves measuring the length of the DNA fragment and determining the specific alleles present in the DNA sample. This information is turned into a DNA profile which can be used for identification purposes.
Fun facts. Dinucleotide repeats are the most common short tandem repeats in the human genome. Prior to the 20th century, STRs were referred to as junk DNA until scientists began to discover their uses in the body as well as forensic uses. STRs can cause frame shift mutations that can mutate bacteria into becoming more contagious. STRs are also a part of regulating transcriptions, the process of making RNA strands in DNA. Most importantly, they are extremely useful in the construction of genetic maps, which can lead to aiding in linking individuals in paternity tests. Thank you for watching. See you next time.